So in this congress, we have shown that lurbinectedin has a very interesting activity in patients with a BRCA mutation and metastatic breast cancer. So patients with mutations in BRCA1 and 2 share homologous recombination deficiency. And they do have difficulties in repairing tumors that have double-strand DNA breaks. Lurbinectedin is a trabectidin analogue that specifically binds to the CG-rich motifs and inhibits active transcription of genes that are at that time being actives and being transcribed. By doing this, it then causes and induces the formation of double-strand DNA breaks, and which finally collapses the replication forks. That has also shown to be specifically interesting and active in cell lines that have homologous recombination repair deficiency. And that led us to investigate the role of lurbinectedin in this group of patients with metastatic breast cancer and a BRCA1 or 2 mutation. Right. So the trial met its primary endpoint. Overall response rate was 40.7% in the overall cohort, with a median progression-free survival of 4.1 months, an overall survival of 20 months. When looking at the specific clinical characteristics of these patients, BRCA1 mutations responded in 26%, and BRCA2 mutations responded in 56%. We also observed differences based on prior exposure to platinum, with patients who had not been exposed to platinum a 56% response rate, and patients who had been treated with platinum a 26% response rate. But we did not observe differences according to hormone receptor status. Median duration of response was 6.7 months, and we do have long responders, 7 patients who had responded for more than 10 months, and with a very safety profile. The trial was initiated at 7 milligrams flat dose, but then in order to improve safety, it was amended and adjusted for body surface, and it was adjusted to 3.5 milligrams per square meter every 3 weeks. The safety profile was very positive, with only neutropenia grade 4 in 5% of the patients, febrile neutropenia grade 3 in also 5% of the patients, nausea and vomiting grade 3 in 5% of the patients, and no alopecia, no mucositis, no common adverse events that we usually see in other compounds. In addition, preclinical evidence that lurbinectedin has tumor activity in PDX models that are resistant to PARP inhibitors, and actually we saw responses in 75% of them, prompted to an expansion cohort, and now we are specifically investigating the clinical activity of lurbinectedin in patients who have been exposed to PARP inhibitors. Right. So our interest is that since these new novel agents are now being used or are being tested in patients with BRCA mutations, we wanted to actually analyze what's the optimal sequence of treatment for these patients, considering the kind of mechanisms of resistance that they may develop when they progress to these agents. So our preclinical work has shown that 75% of our resistant PDX models to PARP inhibitors respond to lurbinectedin, and we now want to test this as an expansion cohort in our phase 2 trial. Lurbinectedin, I believe it's being tested as a phase 3 trial in patients with ovarian cancer resistant to platinum, and in combination with doxorubicin as a second line in small cell lung cancer. So I think our conclusions is that we have shown that lurbinectedin is active in this phase 2 clinical trial in patients with metastatic breast cancer and BRCA1 or 2 mutations, and that these results encourage further development of lurbinectedin in this specific population.